drug delivery, and neurodegenerative diseases like Alzheimer's disease, Parkinson's, and traumatic brain injury. Notably, he had developed a pioneering concept using plasma change as a therapeutic approach to reduce amyloid deposition in Alzheimer's disease. Dr. Ramirez's work reflects his dedication to translational research and advancing innovative treatments for neurodegenerative diseases, including the aforementioned plasma change, mitochondrial transplantation therapy, in vivo reprogramming, and more. Thank you very much, Dr. Santiago Ramirez, for being here today, for bringing this amazing symposium, and all the stage is all yours.
Thank you so much for the introduction. Thanks to the organizers for having us. It's a real pleasure to be here and share the research we are doing. I think it's a very important step in science, the possibility to transmit and collaborate so we create these networks and better ideas will come up. So I'll briefly introduce these bright researchers. I will do it in Spanish, so I will explain a little bit of the concepts we do to familiarize with the topics. So I will switch now languages. Bueno, entonces lo que nosotros hacemos en este laboratorio se basa en el protein misfolding, el mal plegamiento de proteínas, y cómo este mal plegamiento de proteínas puede llevar a distintas patologías neurodegenerativas. Vamos a hablar un poquito de los conceptos de cómo una proteína que tiene la misma secuencia aminoácidica puede tener distintas conformaciones, y eso llevar a distintos fenotipos en los pacientes, en la clínica. Vamos a tocar un poquito los temas en la enfermedad de priones, que es una de las primeras, la primera enfermedad que fue descrita por su agente causal, siendo una proteína la única entidad que podía llevar esta patología y que podía además ser infecciosa. Vamos a mencionar un poquito de qué pasa en estas neurodegeneraciones como Alzheimer y Parkinson, donde hay un poquito de prion-like, y cómo este misfolding también se puede ver en estas enfermedades. Vamos a mencionar de qué manera este principio de transmisión, de transformación de esta conformación, puede ser utilizado como una técnica diagnóstica, cómo podemos, qué manera esta técnica diagnóstica puede ayudarnos a detectar estas patologías mucho antes de que la enfermedad tenga sintomatología. La primera charla que vamos a dar va a ser en relación a esta técnica que se llama SAA o Seeding Amplification Assay. Dejo con ustedes a Zeina, ella va a hablar sobre este tema. Por favor.
All right. Okay. All right. Good afternoon, everybody. Hola and gracias for having me. My name is Zeina, and I will be presenting on the alpha-synuclein seed amplification assay, also known as the SAA. So this is a novel diagnostic tool for synucleopathies. Just as a bit of an introduction to me, I completed my Bachelor of Science in Biomedical Sciences from the University of Waterloo in Canada, and I'm currently working with Dr. Claudio Soto at the University of Texas Health Science Center in his protein misfolding disorders lab. So just to begin with, as an introduction, as I'm sure Santiago mentioned, we know that misfolded aggregates are characteristic of our neurodegenerative diseases. So a characteristic protein will misfold and cause aggregation. And these aggregations will accumulate and usually become the hallmark of those neurodegenerative diseases. So just as kind of a review and some examples, we have Alzheimer's disease, in which the protein that misfolds is amyloid beta. It aggregates into our amyloid plaques, which are the hallmark of Alzheimer's disease. With Huntington disease, it's the protein huntingtin. And then with the tauopathies, we have misfolded tau that is the characteristic misfolded aggregate here. In prion diseases as well, the normal prion protein will misfold and cause aggregation, which will result in the prion disease. So the focus of this presentation and what will be the continuing scope is actually alpha-synuclein as a protein when it misfolds and creates deposits And well, it aggregates and creates deposits, and we know this best in the neurodegenerative disease of Parkinson's, which falls under the class of synucleopathies. So to begin with, I just want to make sure to introduce this umbrella term. So with our alpha-synuclein monomers, which is a native protein that occurs in the brain, we can have this monomer aggregate and create different strains of pathology. So we have these in the diseases known as Parkinson's, PD, DLB, which is dementia Lewy body disease, and also MSA, which is multiple system atrophy. So different strains will actually create these different synucleopathies, but these are all aggregations of the same monomer, which is alpha-synuclein. And so Parkinson's disease is the second most common neurodegenerative disease, with DLB being the second most common form of dementia after AD. And MSA is also a very prevalent synucleopathy. This, combined with the fact that 40 to 50% of Alzheimer's patients also show alpha-synuclein deposition, makes it really important for us to have an objective and biochemical assay that can more accurately and can diagnose these diseases early on in their pathology. So to kind of explain, we basically have talked about how the aggregates are characteristic of these diseases. But how do these aggregates actually participate in the disease progression? So the disease progression of these diseases, so synucleopathies is what I'll be talking about, but this also applies to neurodegenerative diseases in general, is that we see the clinical onset represented here by the blue and black curves. These actually happen later on in the disease. We can see that the earliest pathological features are actually the aggregation and spreading. So logic follows that if we can detect the diseases at this point, we can do early intervention and have more effective treatments. So how does this aggregation actually begin? We have our native alpha-synuclein, which is the normal form of the protein that exists in the brain. This will misfold, and when it misfolds, it will interact with the other normal proteins and cause them to misfold as well. And these are stabilized by their protein-protein interactions, and they form these oligomeric species, which we will refer to as seeds. Now, these seeds will recruit more alpha-synuclein and basically elongate into fibrils. This elongation process is also known as nucleation. So we can call this process seeding nucleation. These fibrils can then aggregate further, and they will deposit intercellularly, and we know them as Lewy bodies. Now, these Lewy bodies are present in different areas of the brain and follow a pattern of prion-like spreading. The regions of the brain that these aggregates can congregate in or are most concentrated in is dependent on the type of synucleopathy that we're looking at. So now that we know that the aggregation is the disease progression, and we have aggregation involved, the question is, how do we actually detect those early aggregates with specificity and sensitivity? So the way that we do that is we look at what the mechanism, in depth, what the mechanism of the seeding nucleation process actually is. So what we did in our lab is that our strategy to detect these aggregates in the brain was to harness the native seeding nucleation process that happens. So to go a little bit more in depth in it, we have our protein that is normally folded. It undergoes misfolding and is now our misfolded protein here. That misfolded protein will interact with more protein, causing them to misfold, and then we will get this oligomeric species that is stabilized by those protein-protein interactions. Now, once we have this seed being formed, we have that elongation phase, which is the nucleation. And this will continue to recruit the alpha-synuclein and cause these large fibrils and aggregates. So this is a graphical representation of it. And so this is what happens in the seeding nucleation process in the brain. So now what we did is that we took a biological sample from a patient suspected to have seed, and we added it to normal protein. And what we saw was a very large increase and acceleration in the aggregation process. Now, this process in the brain will normally take decades to occur for this aggregation to reach that point. So we needed a way to essentially So basically what we did then was that we took biological samples from patients that had both that had no seed, so the patient here no seeds, and then patients with seeds. So what we did is that we introduced a biological sample to normal protein. If the patient had no seeds, we would see a very slow rate of aggregation, which can be seen by this curve in the graph here. Now, if the patient sample did contain seeds, we saw aggregation occurring very, very, very quickly. And so essentially, we wanted to harness this. And our strategy is to harness this ability of misfolded oligomers to seed the polymerization of this monomeric protein and enable a very high sensitivity detection. So, because this process takes decades to occur, we needed a way to speed it up, to amplify it so that we can actually see it in vitro in a realistic timeline. And so what we did is that we took that biological sample that contained seed, we introduced it to normal protein here, and then we had that elongation, that nucleation process occurring. But what we did that allowed this to speed up dramatically is that we mechanically fragmented it. So when you mechanically fragment it and then let it continue to elongate, you will get an exponential rise in these seeds because that fragmentation elongation process was cyclically repeated, which is where that amplification comes, and this is that cyclic process. So that's very key to this, that those cycles continue to repeat. So we wanted to be able to optimize this technique for alpha-synuclein specifically. So what we did in our lab is that we have previously worked with other proteins and applied the same kind of fragmentation process, like the seeding amplification process to them, but we wanted to be able to optimize it for the alpha-synuclein protein. So what we did is that we grew alpha-synuclein in bacterial culture, we purified it, and then we created oligomers and seeds. And then we adapted the SAA to ensure that if we didn't have any seed present in the sample, illustrated by this black line here, we would have no aggregation occurring. However, when you do add seed, which is indicated by these colored curves here, you do get aggregation. And with the increasing concentration of seed, you get faster aggregation. So here we can show that this is a highly sensitive technique because if the seed is not present, we do not get any aggregation. Now, how do we show that it is sensitive, that only alpha-synuclein is going to be aggregating? Well, to do that, we took other seeds that will exist in the brain in relation to those neurodegenerative diseases that I told you about before. So we took purified A-beta, purified tau, and then our purified alpha-synuclein. And what we were able to see is that when we added all of these seeds into the SAA, only the alpha-synuclein aggregated. So that basically shows that not only do we have a specific SAA, a sensitive SAA with only the alpha-synuclein seeds aggregating, it's also sensitive to alpha-synuclein only because these other seeds that are present in the brain will not aggregate. And so this technology was established about 8 years ago, and in that time, our lab and other labs have done a lot of work with this. So due to the time constraint, I won't go over everything, but I'll summarize basically some of our publications and results to illustrate what this technology can do. So this is the first paper that implemented the SAA, and what we did here was that we used cerebral spinal fluid from patients who had synucleinopathies, so PD, MSA, DLB, and we also had control patients present. When we used the alpha-synuclein SAA to diagnose these patients, we were actually able to distinguish the synucleinopathies and the controls with very high specificity and sensitivity. So we were able to show that the technique that we had established in vitro also works with these patient samples. And so now we have to show that this is actually reproducible. Can people besides us do this? And we were able to show that with this study here. So this study essentially occurred in 2 different labs who used 2 slightly different methodologies, but they were both based in the seeding amplification SAA. And so both of these labs used the same samples and they were blinded. So the researchers conducting this did not know whether the sample was from a patient with synucleinopathy or not, and they achieved the remarkably very similar results. So this essentially shows that, okay, this technology is reproducible. And so here we can see, I just want to clarify, you will see the word PMCA and RT-Quick. So I've been referring to the technology as SAA, or seeding amplification assay, and that's because that is the new unified umbrella term. Previously it was known as alpha-synuclein protein misfolding cyclic amplification, which is the fragmentation elongation technique I had described, and RT-Quick. They both are based in the same principle. It's just all known as SAA now for more convenient nomenclature. So this next study was similar to the previous one, but this was done in actually 3 different labs, and we were able to again see those same results where those samples that were blinded were able to achieve very similar results, showing the high specificity and sensitivity. Now, this study was essentially valuable because it was able to clinically validate the utility of our SAA. So what happened in this study is that it involved a very, very large cohort of over 1,000 well-characterized synucleopathy samples. And what happened was that these patients, all with various Parkinsonisms, when we ran their samples through the SAA, we were able to essentially accurately diagnose. So the clinical diagnosis and the SAA results fully matched up. So this study was very valuable for us because it clinically validated our technology. So with what we have discussed so far, we've essentially been able to show that detecting aggregates at the point of clinical diagnosis is possible, it is accurate, it's very specific, and it's very sensitive. But if you remember that first graph that I showed you, clinical diagnosis and clinical onset occurs much, much later, decades later, than when aggregation starts to begin. So we wanted to investigate whether or not we can actually see the aggregation before onset of symptoms. And so we were very fortunate to be able to work with a group that had samples of IRBD patients. What is IRBD? Well, IRBD is a rapid eye movement sleep behavior clinical condition, and the significance of this condition is that it is prodromal to PD. That means that most of these patients who have IRBD will develop Parkinson's later on in their life. And the group that we collaborated with, they had samples of these patients before they showed any clinical symptoms of PD. So we had samples before clinical onset and also after clinical onset. And so these lines here represent different patients, and we were able to show that a sample of a patient's CSF sample from 6 years before they actually showed any clinical symptoms of PD was positive for SAA. So when that IRBD patients undergo phenoconversion into Parkinson's, we can actually detect that way before. And unfortunately, we had a limited availability of these samples, so if samples were collected perhaps even 10 years before, who knows, we may have been able to see aggregation even then, but based on what was available, we were able to see that our SAA can pick up on those alpha-synuclein aggregates before clinical onset occurs. And so now that we have essentially developed, validated, and also shown some preclinical diagnosis of these patients, we wanted to be able to see if we can adapt this SAA to not only distinguish synucleopathies from like non-synucleopathies, but if we could actually distinguish between synucleopathies. So what we did was that we were able to adapt this SAA to distinguish between PD and MSA, which are both synucleopathies. But like I had mentioned earlier, these are different strains of synucleopathies. So this SAA can actually distinguish that the structure of these two synucleopathies are different. So in conclusion, we were able to develop this SAA by showing that by harnessing the alpha-synuclein aggregation mechanism, that seeding nucleation mechanism. So basically, alpha-synuclein aggregation occurs in the brain before symptoms and clinical onset. We were able to develop a very highly sensitive and specific technique that was able to detect these aggregates using our seeding amplification technology, and this allows for more of an objective and biochemical diagnosis of this disease. And our SAA, as a technology, we were able to validate through reproducibility and very large cohort studies that were blinded. We were also able to show preclinical detection, and we were able to adapt it to differentiate between synucleopathies themselves. And so my colleague Danielle will go over more of the distinctions and differences in the structure, but I was just basically introducing the technology that allowed those samples to be analyzed for that. And so that concludes my presentation. I would like to acknowledge everybody in our lab. This is a picture from our retreat earlier on this year, and this is Dr. Soto himself. And then also I would like to acknowledge our funding partners and organizations that were able to make this research possible. Thank you.
Any questions?
there's one up there.
Hola. Well, I have only one question about your technique, and you say that the amount of the concentration of the aggregate seeds in the preclinical symptoms, the patients with preclinical symptoms, but in the late stage of Parkinson's disease, this amount of or concentration of aggregates have a difference?
Yeah, so essentially the way that the aggregation occurs is that earlier on in the disease you will have like less aggregates, but as time goes on you will have like more aggregates occurring. But the idea is that our technology is an amplification technology. So the point is that even if like a minuscule amount of seed is present, we will be able to detect it because we're amplifying what is there.
But not the stage of the patient.
If I'm understanding correctly, I don't know. We can't use this as this technology to say which stage of disease the patient is in. No, not yet. We are working on some quantification techniques to be able to actually determine that in the future. So that is something that our lab is working on. But currently, as the technology stands, we're not able to stage the disease with this technology.
Okay, thank you.
No problem.
Okay.
Any
Another
other questions?
question? Okay, continue.
So the next researcher is Danielle. She will be presenting what else we can do with this technology, not just diagnosis, what allow us to do. So please, go ahead.
It's a good start. Okay. So
Do you have the PowerPoint?
the PowerPoint.
They're working upstairs. ¿No se escucha? Ahí está. Ah. Bueno, para completar un poquito la pregunta que tú hiciste en un momento sobre si podemos detectar en base a la cantidad, de hacer como una correlación con el stage. Lo que estoy haciendo ahora es un experimento usando un modelo animal donde nosotros inducimos la agregación inyectando fibras de MCA en el cerebro. Y nosotros ya sabemos cuánto eso va a durar. Entonces lo que estamos haciendo es tomar longitudinalmente muestras de CSF en distintos grupos de animales y lo vamos a correlacionar con la progresión de la patología para tener un estimado en el modelo animal de cómo podemos, o qué tan temprano, qué tan antes podemos detectar ya tanto en relación a la patología, en la histopatología como en la signología clínica. Entonces, estamos trabajando en eso también en los modelos animales.
Oh, there we are. Okay. Okay, so hola. Thank you everyone for having me. I'm enjoying being in the city so far, so excited to be around here. My name is Danielle Harrison, and today I'll be talking on the use of cryogenic electron microscopy to investigate structural aspects of amyloid filaments associated within different neurodegenerative diseases. So here I'll go from like a broad view, and I'll narrow it down to a microscopic view of what is a causative agent in neurodegeneration. So a little about my background is I received my Bachelor's of Science in Behavioral Neuroscience at St. Edward's University in Austin, Texas, where I developed a passion for neural mechanisms and what occurrences can cause actions to occur. After that, I have started work in the Texas Medical Center in Houston, Texas, where I work at the University of Texas Health Science Center with Professor and inventor Dr. Claudio Soto at the Protein Misfolding Disorders Lab. There, I do structural studies of the misfolding proteins. So to begin with, so that we're on the same page, I will briefly describe what structural biology is, which is the field of studying the arrangement of molecules. So within proteins, that'll look like 4 structural levels. We have primary, the amino acid sequence forming the polypeptide chain. Secondary structure is the 2D appearance in alpha helixes or beta plated sheeted. Tertiary structure is the 3D structure that you can see. And quaternary structure is the interaction of 2 subunits and their engagement. So each of these can be studied in different manners and with diverse goals in mind. The very first accomplishment with structural biology is this structure here. This is DNA. It was first discovered to have a double helix conformation in 1953. So it was done by Crick, Watson, and Franklin with the method of X-ray crystallography to identify the shape. And with this method, they were able to get the structure, which beforehand, it was not known how DNA was able to function the way it is. And with the structure, they were able to elucidate the mechanisms that was enabling it to function. So after this, structural biology really took off, and shortly, 2 years after, protein studies started with the many different conformations that proteins can take. Different methods are, 1, X-ray crystallography, which is the first one, the oldest, and it's still in use today. Electron microscopy is a tool that's used in structural biology that I'll speak a little bit on. Nuclear magnetic resonance, NMR. Cryogenic electron microscopy, aka cryo-EM, is the main one that I'll be speaking on. These different methods, they all have their benefits. They have great use. So NMR is good at elucidating the structure of small molecules, but it is not able to glean information from larger protein complexes. X-ray crystallography is able to view the larger molecules and also the small molecules, both enabling high resolution to get atomic placement. Cryo-EM has recently developed into being able to work with large protein complexes as well as small molecules now, with the advancements that it's had to its electron beaming source and the optimization of sample processing, as well as it's a highly computerized process, and we've had recent advancements in the last 10 years to enable the high resolution structure to place atomic models for small molecules and large protein complexes. So light microscopy is also a great tool, but it only gives you the cellular view of live imaging, but it's not able to bridge the gap into atomic placement, where cryo-EM is at the cusp of viewing larger molecules. So to get further into how cryo-EM works, as the name suggests, it's cryogenic. a large part of it is the freezing process. So this is the automatic system that we use to freeze our grids. And it's important to, it's really important to the process because without the freezing part, the sample wouldn't be shown in its native state. So NMR and crystallology, they have the other benefits, but you're not able to ensure that you're seeing a protein in its native state as you are when you flash freeze for cryo-EM into, by placing your sample on your grid, and then flash freezing into liquid ethane, which is liquid nitrogen temperature, but it happens way quicker than liquid nitrogen is able to do. And so that'll be in the center there. And this is what the scope looks like. It's very large. This is an internal view of it. So your tiny sample that's placed on a grid will be placed in here. And this is the image processing center where your initial computation will take place, where you can fix the dose of the electron beam that you are shooting onto your sample and choose images of your sample throughout the grid. So this is what the grid view would look like from a low magnification view. And then inside the grid, you'll have a grid square. Further, you'll have a grid hole where your sample is located. And you'll have many pictures taken of your protein, 20 to 40,000 typically that we deal with. And once you have these images of your protein, you can go on to start particle picking, isolating your protein, and creating 2D classifications, which is like the different orientations that you're able to get in images. You can place them in like classifications, so you can have an accumulation of that specific orientation. And in the end, you'll have the many different views that are able to be stitched together to create a 3D image of your protein. So you do 2D refinement with that, and you can create a final 3D reconstructed model of your protein. So now I will get into misfolding diseases and how this is used there. So to start with, misfolding diseases is, as my colleague spoke about, both of them, is a conformational change that occurs to a cellular protein. So we have these proteins, and they're functioning normal in our bodies in a healthy individual. This is what a cellular protein would look like for prion, with the C-terminal having alpha helixes. When disease occurs, the misfolding will cause the protein to take beta-sheeted formation in that same area where before it was alpha helixes. So now you have beta sheets where the alpha helixes were and throughout the protein. And they're intermolecularly connected, forming these beta sheets. And as the disease states rise, the amount of this protein also rises with it. The pathological properties of prion and prion-like protein aggregates in misfolding diseases look like this in the brain images. So Alzheimer's plaques for amyloid beta are here in accumulation, and tau tangles are represented like this. Prion plaques are associated here. Parkinson's Lewy bodies are formed by the alpha-synuclein aggregates, and they're formed here. For ALS, in this image, this is SOD1 forming an aggregate. And in Huntington's disease, this is Huntington forming an aggregate. So all of these, when you look at it in transmission electron microscopy for negative staining, they all look the same. They have the same fibrillar structure. So to identify more structural features to get the architect of these proteins, we have to do further structural biology studies.
So we use cryo-EM. And to begin using this process, we have to do purification, and that's different depending on the protein. We'll use bacterial expression, or we can purify from brain tissue. And then we will check quality with electrophoresis and negative stain EM to confirm that your protein is there and there's minimal contaminants of not your target protein. And then you can do data accumulation where you take your grid with the sample placed on it and go for cryo-EM imaging and imaging processing to, in the end, after refinement, get a density map and a final structure. So you're only able to get that high resolution structure of atomic placement in your protein if every part of this process is optimized for single particle analysis. There are many misfolding proteins in human brains. These are some associated with neurodegeneration. Amyloid beta is associated with Alzheimer's disease. Tau associated with Alzheimer's as well as other tauopathies as CTE, Pick's disease. Alpha-synuclein is associated with Parkinson's and other synucleopathies. TDP-43 is associated in ALS, and PRP is involved in GSS, Kuru, variant CJD for human diseases, and they're also affecting animals for prion diseases. So within these different diseases, there are some proteins that are causing different clinical symptoms in patients. So you'll have tauopathies, which are associated with Alzheimer's, CTE, Pick's disease, but with different clinical symptoms. And with that, when you look at the structure that has been previously published, you can see that it has different folds that are forming as well. And alpha-synuclein, as the protein also takes different conformation in an MSA and Parkinson's disease. So we were looking at, with the SAA, we were able to have a large amount of material. And so this is SSA-generated alpha-synuclein from MSA patients. And after several years, we were able to get this structure of the electron density map right here of alpha-synuclein. So these are two protofilaments creating this density map, and you have the atomic model with the beta sheets as well. So when you compare this to the two protofilaments to the published MSA structure, you can see that they overlap, the core regions are overlapping. So it has an RMSD value of 2.6 here of similarity and 1.6 here. So the SSA that is generated by our lab for amplification and diagnosis techniques is showing that it is the same structure being produced in patient brain. And this makes it a viable use for being able to do further studies and preserving patient sample to do mechanistic studies. Another protein that I deal with is prion, and it's associated with an animal disease, chronic wasting disease, that mainly affects cervids such as reindeer, moose, elk. And we are looking here to see what is the causative agent of CWD and what that looks like in its atomic placement. So this is the structure from a natural case of CWD of prion-infected elk brain that we were able to get the structure of. Well, so it just showed you the beta sheeted structure and how it's throughout the protein. But this is the top view that you can get its spiral fold. This is at 4.6 angstroms, so you can start to see the beta sheets at that point. But yeah, and yeah, so that's the top view of it.
It's not available.
It's okay, it's okay. So I also showed how it changed from the known structure of uninfected elk brain into that structure, which is very different from the alpha helixes to the beta sheet. So the implications of knowing the structure is being able to glean, being able to do mechanisms of action. You know the structure, you know what the structure was before, and you can now target that. And that goes into drug discovery. We can do structural-based target identification with ligand-based screening and structure-based screening. And both of these are in review for Nature Communication right now. So I would like to thank you all for listening. This is my lab, and I would like to especially thank everyone in red for their contribution to the projects that I have mentioned here, and everyone else for their support. Thank you. Any questions?
We have a question over there.
Aquí está.
Sí, claro, claro. Claro.
Hay dos preguntas.
Ajá.
La primera para Aina, creo que así se llama. ¿Hay algún otro método de detección, por ejemplo, no sé, con anticuerpos, en donde los mismos anticuerpos puedan unirse a estas semillas para justamente no hacer esta amplificación? ¿Y qué beneficios o qué ventajas tiene el método de amplificación que ustedes utilizan en comparación con utilizar, por ejemplo, anticuerpos o algún otro método de detección?
Okay. So the question is if there is any other method, antibody-based, like ELISA or Western blot, that could be used to detect these seeds in fluids,
Yeah.
and what are the benefits of this amplification compared to those?
So basically the amount of seed that is present in the samples that we did, if we just ran them as is using those other techniques, we wouldn't be able to see anything because it's a very small amount. Now, if you have a brain biopsy, but with that you can technically homogenize and run a Western blot and be able to detect the alpha-synuclein aggregates, but obviously that is incredibly invasive. So that's why we do the amplification, because the amount that is available in the samples that we have access to, which is like cerebral spinal fluid, with those other techniques it wouldn't show up, and then when we amplify it we can see it. But if we were to, the other real biochemical technique to be able to detect it would be essentially a brain biopsy, where we would take the brain homogenate and then run it based on the biochemical techniques that exist. That's why this technology is so valuable, because we don't need to take that brain biopsy.
Ok, de acuerdo, gracias. Y la segunda pregunta es para Daniel. Veo que estas proteínas mal plegadas tienen una coincidencia, una homología en la proteína, una homología amplia. No sé si sea del 100% en todas estas proteínas, y si es posible utilizar algún tipo de ubiquitinación o algún método para marcaje, como para empezar a degradarlas si este sistema ya está un poco desequilibrado.
Si es que entendí bien la pregunta, menciona homología entre las distintas proteínas prion-like, entre distintas patologías, o dentro de la misma, dentro de la misma proteína entra las distintas patologías causadas por la misma proteína?
Ambas cosas. No sé si entre distintas proteínas mal plegadas de distintas patologías guardan alguna homología, y también si la misma proteína, porque bueno, entiendo que dentro del proceso de síntesis de proteína, cuando estamos hablando de una GABA, pues todas las GABAs tienen pues la misma homología, salvo que hay algún polimorfismo, ¿no? En este caso, inclusive dentro de la misma proteína, ¿se presenta una alta homología en estas proteínas?
So, this question is very molecular based. So, when you synthesize proteins, you will have certain homology. And so the question is, if there is, knowing these homologies or how these homologies share between these misfolded proteins, if there is any methods like ubiquitination.
Which one?
Ubiquitination. I can help you. I help you with this because it's out of the topic of the structure. So if you allow me, I can answer here.
Okay.
Okay. So this is outside of the structure. So basically, the homology is almost 100% in the alpha-synuclein between MSA and PD. The only difference is how the conformation changes. So the problem is, based on the strategy of targeting, do digestion by targeting with ubiquitination, there is a technology now called PROTAC. You create basically a chimera with a E3 ligase. Then you can target these specific proteins and take them to the proteasome for degradation. The problem is that these aggregates are so big that you will overwhelm the proteasome. And even if you are able to send it to a proteasome, you will be creating more seeds. So you can have an opposite effect. That is because the protein is so stable, it's so stabilized, the amyloid formation, that you will have protease resistance. So there is a core that you won't be able to degrade. And that core may have seeding properties. So if you overwhelm the proteasome, then you will create basically what we are doing in vitro by shaking or by sonication, disrupting these fibrils. You will be doing almost similar procedure. So it's kind of difficult. What has been done is, I know that there is PROTAC for other proteins that play some roles in promoting the progression of the disease that you can treat with this targeted degradation. But for amyloids, it's very difficult. For A beta, for instance, it's an extracellular protein, so it's even more difficult. So you won't be able to bring it back inside whenever it's already secreted outside of the cell. So we have discussed that approach, but it's complicated to do it biologically. The system won't allow it too much. And these big companies, they're 100% focused on that method. They already know. They are not even making efforts to do it. Yeah.
Bueno, hola, y bueno, sí quisiera, bueno, tenía dos dudas, pero la segunda era pues sí más o menos relacionada a lo que ya respondieron de si se podía ver como una dinámica pues molecular, o sea, de la estructura y ver si se podría degradar, pero bueno, creo que ya con lo que dijo pues se responde bastante bien. Y la otra es de que qué sucede en sí con las proteínas que se amplifican, o sea, cuál es su función o sí, qué es lo que sucede una vez de que esas como semillas se amplifican.
Okay. So this question can be for any of you.
Okay.
So it's what is the role of these proteins when they get amplified, the misfolded get amplified? So if you are doing the SAA, so why we need or why we amplify this misfolded?
Mm-hmm.
What is so relevant for the process?
Yeah, so I think maybe.
Sí, bueno, supongo que es O sea, sí, para el proceso como de identificación de las proteínas, ¿no?
Ajá.
Pero una vez de que se hace más grande la proteína, o sea, no deja de, al final de cuentas, ser como esa proteína como patológica, pues, o sea, porque cuando se crece ya no apunta, pues, como para ser de Alzheimer.
So, cuando tú el concepto de amplificar es similar a lo que pasa en el PCR, ¿no? Tú tienes un template y lo vas amplificando y vas creando múltiples copias de eso para poder detectarlo, porque de otra forma no podrías detectar en una electroforesis. Entonces, en este caso, el objetivo de amplificar es netamente aumentar la señal para poder detectarla. En el paciente no queremos que eso pase. En el paciente lo que queremos es evitar que la agregación ocurra. Y para eso es lo que Daniel estaba proponiendo, que si tenemos la estructura, podemos desarrollar, hacer estos estudios de molecular docking y buscar cómo evitar que esta agregación siga pasando. Y como tenemos el SIA, podemos hacerlo in vitro, podemos hacer screening de múltiples compuestos para ver cuál de esos compuestos inhibe la agregación y luego llevarlo a la clínica. Pero en el fondo nosotros no queremos promover la agregación in vivo en el paciente.
Okay. Sí. Muchas gracias.
Another question?
Por acá.
So, I got a question about the topic of you're talking about, and well, it's from both of them. It's about the molecular docking in the proteins. So this could be a personalized or medicina personalizada. I don't know how can I say it in English, but it could be a medicina personalizada as a molecular docking. This is because I don't know if the proteins could be different in one patient and another patient. It could has other modifications or in the proteins.
So there is an approach to doing individualized treatment, but with knowing the protein structure, it's the same if you're diagnosed with one disease, that structure is taking place in everyone who has that specific disease.
But it's similar in all the patients? It doesn't be different in protein in patient 1 and the protein of patient 2?
Yeah, so for example, when we talk about like alpha-synuclein aggregation, right, we have, when those aggregates occur, we have those different strains. So those different strains or structures are not actually different between the patients, but between the diseases themselves. So the structures of these, like the structure for the alpha-synuclein aggregate in PD is distinct from the structure of the alpha-synuclein aggregate from MSA. And that last Nature paper that I had shown, distinguishing between PD and MSA, those were run on many different patient samples. So basically in that paper, in that study, we were able to show that, okay, the PD aggregates all behave and fluoresce at one way and they have this structure, and the MSA aggregates all behave similarly and they fluoresce at this structure. And then when we were able to actually show the structure with the paper that's in Nature Communications right now through structural biology, that shows that the MSA aggregate, this is what it looks like in patients versus what a PD aggregate will look like in the PD patients. So there may be like slight differences individually, but it's still distinct enough for us to be able to distinguish those strains.
Okay, thank you.
Sí, si me dejas complementar un poquito, porque hay un lado B también. O sea, puede haber una diferencia estructural si tú tienes, por ejemplo, mutaciones. La A53T, por ejemplo, quizás eso tienes una secuencia aminoácica distinta, podría llevarte una estructura un poquito diferente. Entonces, ahí hay que tener también, dependiendo de cómo se va viendo al paciente o cuáles son las características, considerarlo. Ajá. Claro. Sí. Podría. Claro. Y lo interesante es cómo estas patologías son muy estereotipadas en cuanto a la progresión, y ahí es donde la estructura juega un rol muy importante. Y la estructura está determinando ciertas características. El misfolding te entrega o sea, el gain of toxicity, que se llama, el loss of function también. Entonces esas dos cosas balancean, y tienes pacientes con ciertas características y coincide que el fold es muy similar. Ahora, hacerlo para cada paciente es súper complejo. Por ejemplo, Daniel estuvo trabajando 2 años en este proceso para llegar a la estructura de esta sinucleína, porque en el proceso tú tienes que amplificar, validar tu método de purificación, si es que estás purificando de cerebro, para que la estructura en el fondo siga siendo biológicamente relevante. Entonces nosotros, antes de hacer la estructura, lo que hicimos fue hacer experimentos in vivo. Nosotros obtuvimos el amplificado desde el SAJ, y ocupamos este material para probarlo en células, que teníamos estas células reporteras que tú las agregas al medio y hacen agregación, y tú ves fluorescencia en las células que están agregando. Y también lo hicimos in vivo. Inyectamos este material en animales transgénicos y aceleramos la patología. Entonces, validamos un poco que lo que estamos haciendo nosotros al amplificar en este SAJ tiene relevancia biológica, porque de lo contrario podemos estar amplificando cualquier cosa y mostramos una estructura que no es relevante o no tiene este gain of toxicity. Y eso es lo que el SAJ nos entrega, porque si bien no podemos descartar que existan diferentes conformaciones en un mismo paciente, la que nos interesa a nosotros es la que tiene la mayor capacidad de generar patología, y la que tiene la mayor capacidad de amplificar o tener seeding, y eso es lo que un poco selecciona el SAJ. Con la cinética de agregación tú puedes tener proteínas, folds que son relevantes biológicamente. Entonces esa es la segunda parte que te entrega el ensayo de agregación. Claro. O sea, es similar en cuanto a la exponencialidad, porque en el fondo tú pones una semilla, y luego esta semilla va a inducir el mal plegamiento, y con la sonicación o el shaking se rompe, se fragmenta, y ahora tienes 2 o 4 semillas que van a seguir amplificando exponencialmente. Entonces tiene una analogía al PCR, el proceso. ¿Sí? Lo bonito es que te conserva la estructura original, que cuando tú tienes el agregado al final y haces estos estudios, es lo mismo que tú estás viendo en el cerebro del paciente. Entonces te permite desarrollar una palestra de nuevas técnicas, como, anticuerpos en función a estructura. Para eso necesitas una gran cantidad de material que no podríamos obtener desde el cerebro, y aparte es un proceso muy extenso. Entonces el SAJ te permite hacer todas estas cosas.
Well, I have a question. In this case, you see the protein. Why do you see the protein and not the DNA? Because if you have this protein, it's because it's probably overexpressed, even in Parkinson, Huntington, and all these kind of diseases. But why you study in the protein not all transcription factors and all these kind of things? Because if you sequence the DNA, if you have early Alzheimer, probably, you can see the mutation of one allele that it's probably come to the disease.
Yeah.
1 second.
No problem.
Okay, so we can do DNA sequencing, and that will cover 10% of the cases when you have familial mutations like the A53T in Parkinson's, or the presenilin-1, or the APP mutation in Alzheimer's. The problem is with these sporadic cases, because in there, you don't have any change in the DNA. What happens is you have some other factors that we still are under investigation that will lead to the misfolding of these proteins, and that will lead to the amplification. So we will, if we do genetic analysis in these patients, we won't see differences. We will see the sequence most likely conserved, unless sporadic mutations occur. But where? Because it will be in a neuron, or will be in, it's not like a inherited mutation. So it's not a familial. In that case, yes, we go to DNA and we know that will happen. And that happens for prions too. Prion disease, you have familial prions, and you know that by a certain age you will have. In terms of protein level, in Down syndrome, you have this triplication of this, the chromosome. And unfortunately, you have APP gene in that precise chromosome. So you will have higher levels of APP, you will have A beta, and these persons will develop Alzheimer-like pathology in the brain by age of 40. So yeah, protein levels matters, but in sporadic cases, I mean, protein levels will be normal, genetic, I mean, there won't be some mutations, and could be other multiple factors like ApoE genes, ApoE4 isoforms could be influencing the misfolding. So it's very complex to lead the protein. So sometimes DNA is not giving us answers.
Thank you. If you have in the transcriptomic part, you can see something, or only is when you have all the protein, when you have the protein? Because if you say that you can see in DNA and you can see, well, you can see in protein, but in the middle, in RNA part, like transcriptomic part, you can see something, or no?
So we are doing two approaches for transcriptomics. We are doing single cell sequencing. The problem is that you won't see changes so early, and that is what everybody wants to do. Try to find biomarkers and precise genes. So there is some, but it's like a cluster of I don't know, 20 different genes that could correlate to the progression. So it's a bit challenging. Then the other approach we are trying to do is using transcriptomics, spatial transcriptomics, which is transcriptomic data in the tissue. So you can localize gene expression, gene changes in the mRNA at different brain regions. So you can associate those gene signatures or mRNA signatures with amyloid plaques or with tangles. So that can give us a little bit, but it's always after the disease happened. It's not like before, because we will not have access to that tissue. So it's challenging. Animal models, they gave us some information, and we are trying to do some experiments using iPS cells to try to like incorporate human cells, a small amount of human cells in a mouse brain, to provide a context of a human genome in the presence of A beta and other neurodegenerative diseases and misfolded proteins, to try to have this information that we cannot have it from a patient that is not already, I mean, with advanced disease or without the need of brain tissue. The other approach we are trying to do is using iPS technology, also developing these brain organoids. So we know we have some familial mutations, so we grow the organoids. They will have a high expression of APP. They will have some A beta. And from there, we could get some mRNA information. The problem is we don't have immune cells. We don't have neuroinflammation. So that limited, everything is a model, will give us small answers, but we will never get to the main answer. So it's what everybody wants to do in the transcriptomic, but it's very challenging to get to that point.
Okay. Thank you.
Bueno, si ya no hay más preguntas, les pido un fuerte aplauso para nuestros expositores. Y pues me gustaría recordarles que nos acompañen a lo que continúa el programa, que es la actividad cultural que va a estar aquí afuera, y posteriormente a la última conferencia, y a registrar su asistencia, por favor. Gracias. Un fuerte aplauso.